Okay, let's get started. So my name is Ron. I'm the editorial director here at JoVE. So thank you everyone for attending today. And today we have André Gerber. So he published a paper with us in 2018. The title of the paper is Oligo-based Tandem RNA isolation procedure to recover eukaryotic mRNA-Protein complexes. So this was a very well received article at JoVE, and so today André will be talking about some recent developments to the technique over the last 5 years. All right, so André, I will leave it over to you.
Yeah, thank you very much, Ronald, and thank you, JoVE, for inviting me to give this webinar. As Ron mentioned, so I will talk about a tandem RNA isolation procedure to recover RNP complexes. Now, to give you a brief overview, as you all know, messenger RNAs or every RNA is not naked in the cell, as shown here. It's always covered by a host of proteins forming so-called ribonucleoprotein complexes by association with RNA binding proteins. In addition, various non-coding RNAs, such as microRNAs, also assemble via RNA-RNA interactions. Now, these whole complexes, in particular RNA binding proteins, guide the fate of RNAs and of mRNAs, here shown, for instance, translation. So to bring complex into translational machinery, to the ribosomes for protein synthesis. The complexes can be stored or localized to subcellular regions, and eventually the RNAs are decayed. Now, to control, it needs all these RNA binding proteins, so it's of an imminent importance and interest to develop experimental approaches to identify cellular RNA-protein interactions. So to date, there are different approaches on the market, I could say. So one is here shown on the left, involves RNA aptamers, possibly the oldest approach, where you express your RNA with an aptamer, like a short RNA sequence that can interact with a ligand that can be an antibiotic, but also a protein, like you may have heard about the MS2 protein, and you use that to recover your target RNA for further analysis. What we're mainly talking today is about antisense oligo-based techniques shown here in the middle. In this approach, you cross-link the RNA-protein interactions in vivo by UV or chemicals, and then you purify your RNA, specific RNA with antisense oligos that anneal to your target RNA, and you recover the complex with streptavidin beads that interact with the biotin for further analysis. And this will be the major topic today. I just want to briefly mention this as to the right, which is in-cell labeling or proximity labeling technique that has recently been developed in the last years. And basically, it involves a modified CRISPR-Cas system where you target the bindylation enzyme to your target RNA via guide RNAs, and then you grow your cells in the presence of biotin, and the biotin will be incorporated in the vicinity of your target complex, and you can purify these proteins then with streptavidin beads for mass spec analysis. Now, coming back to antisense oligonucleotide approaches, there are a variety of approaches. I have here compiled some in a recent review on the topic. So most of these procedures involve short DNA or modified RNA molecules coupled to a biotin moiety at the 3' or 5' end, and involve a cross-linking procedure. And as you've seen, several non-coding RNAs have been recovered with this method. However, we focused on TRIP to have something at hand to particularly focus on messenger RNAs, and we have shown that, for instance, in that paper that Ronald mentioned. Now, the TRIP, how it works, there are several steps I will go through subsequently here. So first of all, as I mentioned before, you try to cross-link the RNA-protein interactions in vivo or in cellular by using UV radiation. Otherwise, you can also use a chemical cross-linker like formaldehyde at low concentrations, like 0.3 or to 1%. So we have your cross-linked RNA-protein interactions. Then you follow with a cell lysis. And you can now purify in the first step. We purify all the polynucleated RNA, which includes most of the mRNAs, via oligo-DT beads. So these are standard beads you can buy. And the good thing is here, since you have cross-linked the RNA-protein interactions, you can use very stringent conditions to purify that to remove all the unspecific interacting factors. And the second step then, if we have this pool available, is to capture selected RNA with antisense oligos, with a biotin moiety, with streptavidin beads. Now, the advantages we thought about this two-step is that in the first step, you remove every kind of unspecific biotin binding proteins, because this does not involve a biotinylation or streptavidin beads. And secondly, you also remove many non-coding RNAs, or like ribosomal RNAs and tRNAs that could obstruct your results, because mRNAs is only 1.5% of the whole RNA population in cells. However, you can also apply the second step separately, but in that case, it would be advised to pre-clear your extract with streptavidin beads. Now I will go through the steps individually. So one thing is the target design. So we use the antisense oligo, which is 19 to 22 nucleotide long. We use 2' O-metal modified RNA. So we use a modified RNA because the modification increases the stability of the probe and makes a stronger interaction with complementary RNA sequences. Also, if you use DNA, you have to be aware that you could have RNase H activity and digest. Then most studies are like we use the biotin tag at the 3' end or 5' end and shown here. In addition, you can add a flexible linker that at some distance from the biotin to the RNA that offers more flexibility of your probe to reach the target RNA. In addition, we are testing now an option with a photocleavable linker inserted here, which would allow you to elute the complex just by cleavage with UV light. So just to highlight this modification, so this is the triethyl spacer, the text spacer you can add between the biotin and the oligonucleotide to have a flexibility linker. And what is shown here, this is a recent product we have tested, is this photocleavable moiety, which can be added to cleave off your reagents from the beads. Now, one critical point in this whole procedure is to select the proper probes. And what you see here is the predicted secondary structure of the luciferase coding sequence. And you see it's very difficult here to find a region which is single-stranded to, which is the preferred region where your antisense oligo could anneal. So to find suitable regions, we basically chop off the sequence in smaller pieces, like 150 base pair fragments. So we select some accessible sequences as shown here. These are some examples with 4 oligos we designed to recover the luciferase reporter. So we try to make the oligos with an ATGC content, which is balanced to have a good complexity, so to increase the specificity. And of course, what you should do at the end is a blast search to check for off-target binding, which is always a concern. So at the end, we propose to design 3 to 6 different oligos and different regions of the target RNA, as we have done here. Now, what we usually do and we advise to do is first to do some simple in vitro experiments to test the specificity of the RNA for recovery. And that's shown here for the 4 oligos I showed you before. And we see quite some variability, like some don't work very well, others work pretty well, give you a 50-fold enrichment compared to the input, compared to an unspecific control, in this case, GAP-DH. Here we say like a 10-fold enrichment in this in vitro experiments is sufficient to go ahead. Also, you see here the results from this photocleavable oligo. You basically get similar efficiencies, even a little bit higher. as compared to the default elution procedure. So we elute the complexes by heat. So basically, we just denature the RNA interactions. Yeah, so now there are various factors that contribute to the efficiency and the specificity of the reaction. I just highlight here two. So one thing is the salt concentration. For instance, if you see here, this is if you need a certain salt, physiological conditions seem to be the best one because the salt, of course, has an influence on the melting temperature between the oligo and the target RNA. So what you see here, this is the recovery of a PFK2 RNA. We have tested with different oligos. You see it can be nicely recovered with 150 millimolar salt versus a control, which is the ortholog PFK1 mRNA, is not recovered in this experiment. Likewise, one thing which is possibly more critical than the lookout when we test for every oligo is the washing temperature. Because basically in the principle is that for the annealing, you have a very high temperature at the beginning, you heat the whole thing up to 70 degrees. So then you add your oligo and you let the mixture cooling down to room temperature so that the oligo can nicely anneal. And afterwards, now you wash your sample at different temperatures to remove all unspecific interactions with the probe. And you see that can really make a difference. While with 40 degrees, we see a clear, no big discrimination between target and non-target controls, as shown here for two RNAs from C. elegans, so SEP1 and PGK1, which is a control. But if you increase the temperature, you remove the non-specific binder up to, and you can go. And if you go too high, of course, you go over the melting temperature, you would wash off everything. So it's something you really should adjust for each thing, but usually we are in the range between 55 and 60 degrees. So when you have done this pre-test, you can think, okay, let's go for in vivo samples. This I forgot. This is another highlight. I want to just tell you about off targets. This is really a concern. This is an example we also show in the paper. So this is the example for DNM1, an antisense oligo against DNM1 mRNA, which is an yeast messenger RNA. You see complementary is fully complementary and recovers the rmRNA. However, it also recovers the ACT1 mRNA, although it just hybridizes across 12 nucleotides. So it's really important to be sure that this does not happen. So this oligo was not further followed up and discarded. Now, finally, you can go in vivo, and that's also what we've shown here. So you adapt the procedures in vivo to in vivo, and that was done here. So as shown before, the PFK2 mRNA can be recovered and followed from the input to the first step, oligo-DT, recover it, and with the antisense oligo, whereas the control RNA here, PFK1, which is pretty similar, is not recovered during the second step. And finally, you can do a Western blot of these L weights, and we saw PFK2 protein interacts with the PFK2 mRNA, and this is what we knew from other experiments. So basically, this protein binds to its own mRNA. So in this regard, it's always good if you know some interactors of your target RNA that you can validate this by a Western to make sure everything works as you wish. Similar experiments were done in C. elegans and humans, so I don't go into detail and you can read this up. Now, at this point, we did not do any mass spec, but of course, we have now expanded this analysis, and we are currently analyzing the proteins interacting with different UTRs, so untranslated regions and the 3' end. And as you know, 3' UTRs are a target for RNA binding proteins that regulate mainly stability or translation or the localization of the respective mRNAs. So what we see here are 3 different UTRs fused to this luciferase reporter assays I showed you at the beginning, where we have designed 4 different oligos. We have then selected the best oligos, the best 2. So we did a combination here with oligo 9 and oligo 6 and recovered these complexes from HEC293 cells. Now, in this case, the RNAs were directly transfected into the cells for recovery. We recovered that over the 2 steps. So first oligo-DT selection and then with the antisense oligonucleotide and performed mass spec analysis, and that's what we got. So in total, with all different UTRs, we identified 474 proteins. Of them, a substantial fraction were annotated, and 250 of them are indeed RNA binding proteins. So that's very highly enriched compared to the background, what is present in the proteome. So the method seems to work at the global level. And now what you can do, you compare short versus long RNA. In this case, basically we compared the data, we matched it to the sample without the UTR, which gives you the background level. And we looked which proteins are enriched at least 1.5-fold over this control. And we found 24 or 85 proteins associated with the short or the long UTRs. Of course, this number is much smaller than the total proteins identified across all the UTRs, and this is because we look here specifically, we want to look on changes introduced by UTRs, while here in this pool we have many ribosomal proteins and stuff like that, that of course are on each of the mRNAs. Now, if we look on the data a little bit more closely, we see from the 24 and the 85, they overlap significantly. So these are proteins binding likely to both UTRs, like for instance, poly-A binding proteins. But we also find proteins which are specifically associated with each UTR. So in general, what we can see is the longer the UTR, the more RBPs are associated. That's something you may expect. And we see also common and distinct RBPs which are associated. And of course, now this is the nice basis. You can go ahead and you can correlate these assays with luciferase reporter assays and further studies to look for the functional relevance. So that was just to show you that you can use the method to perform mass spec from small samples. And so it gives you promising results, and we're currently following this up. So in summary, so we have this established strip, but there are now a variety of antisense-based RNA capture methods that have been tested, but the principle is always the same. You have a shorter or longer DNA, RNA, oligonucleotide that anneals complementary and recover the complex with streptavidine beads. In TRIP, we have the addition that we add in the first step the purification of the polylated RNA with oligo-DT beads, which allows you to add more selectivity and specificity by removing unspecific binders. And then in the second step, we use the ASO-mediated RNA capture. Now, what is important, and I think across all studies, these modified antisense oligos need to be empirically tested for efficiency and specificity. Those are the two hallmarks. So how much of the complex, what fraction of the complex you can peel out from the extract or in vitro, and how specific it is. And so one or several antisense oligos can be combined to capture cross-linked RNP complexes from cells and organisms. And here we really saw if we combine, for instance, two oligos, we can, on one hand, increase the efficiency. So you may get more back from the complex. However, it often reduces the specificity because you have more potential for cross-hybridization on specific interactions. And finally, MS-based analysis can be used and integrated with other assays for functional studies of UTR functions in gene regulation. So at the end, I want to thank the people who did the work. So on one hand, Ana Matia Gonzalez and Valentina Yadavea, they set up the assay in the initial phases. And now during the last year, Oliver Rogojski further developed the trip and did these experiments with the luciferase reporter assays. And finally, I want to mention that we have now an opening for a new postdoc position to work on this project and to further do functional studies on it. So thank you very much for attending, and I'm open for questions. Yeah. Okay.
Thank you, Andre. Yeah, so if anyone has any questions, please add them in the webinar chat.
Seems to be everything clear.
Yeah, I think you gave a
Well,
very
I'm
thorough.
sure that I was a little bit as I said, webinars is a special experience. So because you never see the reaction of the people, whether they understood or not, or what are questions. So please feel free to ask any questions where I can ask. Yeah.
Sorry, there is one question.
Yeah.
In the Q&A box. So it's asking what the sample requirements are for this, that is cell number.
Yeah, usually we do that with one big dish. So it's a 115-centimeter dish. But you can also perform this with a lower amount. So it's like 5 to 10 million cells. That should be fine. It's all depending on the efficiency of your oligos. You know, it's also, you see things in vitro and then you go into the cells, it may really change the picture. So you need to have 3 oligos to take further and test them with your cell extracts, because some may super work in vitro, but then in vivo you may have something in vivo that interacts, or this, you know, prohibits the interaction with the RNA that you cannot see in vitro. And also one thing is important is to always think about the concentration of your mRNA in the extract you're using and the probe. So usually probe is, of course, also the probe means the antisense oligo is in a large excess, which is also good. But if it's too much in excess, of course, then you also get more potential for unspecific interactions. And of course, the concentration also has an impact on melting temperatures and everything. So we try to keep the so like in the in vitro experiments, we try to keep the concentrations of your RNA and similar as in vivo, because if you have a much higher concentrated, then it may change. I mean, it's really critical.
Thank you. There is another question. Can you isolate CIRC RNA and interacting proteins?
Isolate what?
Circ RNA.
Circ RNA.
Yeah, circ RNAs. We did not try, but I think that's a very good, that would be very good, because many circular RNAs are very highly expressed. So the better the RNA expressed, of course, the more you get it out and the less materials you need. So I don't see a reason why that should not work. So in that regard, it's worth a trial. Of course, then you would not do the polyase selection in the first step. So the polyase selection is only when you really want to focus on mRNAs, for instance, or other polynylated RNAs. Like some non-coding RNAs also have a poly-A tail, so you could use that here as well. Yes, but it should work. Would be interesting to do, yeah. It's a good idea.
The next question is, how can we prepare the specific modified oligo in your system?
So we order the oligos. All companies, they prepare this, so you can add on the modification 2'-O metal. You add the TEG linker, so we add this linker now by default, and the biotin tag, and also the photocleavable linker is available. However, with the photocleavable, I mean, we thought it's worth trying. The problem here is that if you expose your stuff too long with UV, the RNA starts to degrade. So that may be more suitable for very short RNAs. For instance, IDT, you can order these oligos. That's where we get them from. It's a little bit more expensive than the standard. I mean, other people use DNA oligos. I mean, you can also try, but I mean, as I said, I'm not so I mean, I'm anyway a little bit surprised. Some studies, they use like a whole bunch of different oligos, and it's amazing how that works, because we see so much unspecific interactions occurring if you just use a bunch of oligos. So yeah, anyway, but Mhm.
And one more is there, like we can use this method isolate the extracellular RNA.
Ah, yeah. Yes, that's a very interesting point. Sure. I don't see why you can isolate every type of RNA. You just have to design properly your oligo. You can do that. That would be also a very interesting project.
And the last question is, there is an important difference on the cross-linking method,
Yeah.
UV versus chemical.
Yes. So, okay, so by default, many researchers use UV. And I must say, and this is really a point, it's underestimated. So if you UV crosslink with UVC, so 254 nanometers, you also induce, of course, protein-protein and also protein-DNA interactions, although they are less efficient. But nevertheless, this is somehow notoriously neglected in the field. So you cannot be really sure it's a direct RNA-protein interaction. But in most cases, I mean, it's the best way what you can do. With chemical cross-linking, people are saying, well, chemical you also use to cross-link protein interactions. That's true to a certain state. That's why you should adjust the level of the formaldehyde. So we use very low concentrations, like from 0.1 to 0.3%. By this, you only cross-link really in the vicinity. And if you then apply very stringent wash conditions, I think it's fine. With the UV, another point is also you have to be careful that you don't induce degradation of the RNA. So the UV dose, I would strongly suggest you check that. So you basically do a time course UV of your cells, and then you isolate total RNA and you look whether it's still intact. Because, you know, cross-linker, they indicate how much joule they use. We use 150 millijoule for human cells and 400 for yeast cells. Although there's some variability with the cross-link models, and it should be really looked at. But let's say, I mean, with UV, you are more close to the RNA. With formaldehyde, you can more look on the vicinity. So it's also an advantage. And we just having a study where we compared both. So one side we did UV and the other side formaldehyde. That's in another study. And we see most things are the same. But with formaldehyde, we get more proteins. And these proteins we get in addition are also involve many proteins which are in bigger complexes. So if you want to get a whole complex, let's say, with protein-protein interactions and RNA-protein interactions, the formaldehyde may be the better thing.
All right, so I believe that's all the questions. So thank you everyone for your great questions. And okay, yeah, so thank you, Dr. Gerber, for a great presentation here. And we will follow up with all the registrants after this with a recording of the webinar, if you'd like that for your records. And yeah, thanks again, everyone, and enjoy the rest of your day.
Thank you. Bye-bye.
Thanks again. Bye-bye.